So the fifth cardinal sign of inflammation is given here. Roman physician Celsus in the 1st century AD as Ruber at tumor cum calor et dolore. Okay, so it was the term which was initially used for redness, swelling, heat, and pain. There were four cardinal symptoms or fundamental signs of inflammatory responses. And it is a good thing whenever there is an infection. So you can see if there is an injury or infection, there is inflammation. So inflammation is in fact a kind of sign or to decrease the growth of bacterial cells or other microorganism into that particular wound or site, right? So there were four signs which were included: redness, swelling in that particular zone, heating, that part will be heated, and there is pain, right? An additional mark of inflammation added into in the 2nd century by the physician called Galen. So Galen added one more sign to these inflammatory responses or inflammatory signs, which is known as loss of function. So sometimes what will happen if there is an inflammation, so there may be a loss of function in that particular area, right? So this term was used by the Galen, but it mean in English is actually loss of function. All right. So there are five signs which are related to inflammatory responses. You need to remember these five. Okay. So these symptoms reflect an increase in the vascular diameter that is known as vasodilation. So as I told you, there are some chemokines which will be secreted by these damaged cells, and they will increase they will cause the vasodilation. They will increase the diameter of these blood vessels so that more blood cells or large amount of blood can come into this particular zone where it is carrying large number of cells, right? So because there is increased diameter of blood vessels, resulting in the rise of blood volume in this particular area, and because there is large volume of blood in this particular zone, redness is seen, right, due to increased amount of blood in this area where you are having an injury or if there is an infection. All right, higher blood volume heats the tissue and causes it to redden. Vascular permeability also increases, due to which some of the cells will come out of circulation. The process is known as extravasation, right? So leading to the leakage of fluid from that particular blood vessels. Some fluid or some liquid will be leaked from this particular blood vessels, will accumulate into that particular tissue, resulting in an accumulation of the fluid or liquid known as edema or the swelling. The swells the tissue, which is the another sign of the inflammatory response. Within a few hours, leukocyte also enter into the tissue from local blood vessels. So some of the leukocytes, so major leukocytes are B and T cells, including natural killer cells. So there are three major cells which come under this category: B, T, and NK, natural killer cells, which are also known as the cells of adaptive immune response or adaptive immune system. All right, so up to here you are able to understand that how this inflammatory responses are happening and how the cells are coming out of the circulation, blood circulation, and they are reaching to the site of injury or infection. Now, if you look into the diagram of the major cells, so in the last class also I told you this is very important. So the first cells which will reach at the site of infection or injury are neutrophils, because neutrophils are also present in large number. They are 60 to 70% of all the WBCs present in the blood. So they will reach initially. They will reach first at the site of inflammation or injury or wherever there is an infection in this area, right? Now the diagram is given over page number 34 in your book. Right, so this is a structure of a neutrophil, right? Which is having a multi-lobe nucleus. If you look into the granules, actually they are having two major type of granules. They are known as primary and secondary granules. So these granules are very important because they will contain the enzymes which are responsible for their function. All right. So we will discuss these neutrophils in detail. Uh, First of all, we will move to another PDF. All right. So we will discuss this particular diagram in detail also because because this process is known as hematopoiesis. All right. So this synthesis of different kinds of cells from the same hematopoietic stem cell is known as hematopoiesis. So the same myeloid stem cell, if you are not clearly able to understand this, because this is half diagram of this same diagram which is given over page number a few pages back. All right, so here it is. If you see this particular diagram, this is a complete picture how the different types of cells which are present in blood and lymph are synthesized. All right. So this is a self-renewing hematopoietic stem cell, which is the mother cell, which will give rise to all different kinds of cells, including monocyte, macrophage, neutrophil, eosinophil, mast cells, basophils, platelets, and all. Okay, and RBCs called erythrocytes. Also, the same cell will lead to the synthesis of all the lymphocytes known as T cells. T cells are of two type of populations, T helper cells and T cytotoxic cells majorly. And there are B cells. Okay, and the one major class is called natural killer cells. So these are called lymphocytes, B, T, and NK cells. And the dendritic cells, which are having some phagocytic function and antigen presenting function, are also common to both the lineages, including myeloid and lymphoid. So look at this cell. This is myeloid progenitor cell, which is giving rise to neutrophils, eosinophils, basophils, platelets, RBCs, and macrophages, right? So this cell is shown in this particular diagram now. So this is a myeloid stem cell, which is actually coming from hematopoietic stem cell, right? So as given in this particular diagram. So this is the hematopoietic stem mother cell, which is giving rise to all the cells. And each time when they are dividing, actually one myeloid progenitor cell will be synthesized. And if it is synthesized, each time the other cell which is formed after division is actually hematopoietic stem cell. So these cells are having self-renewing function. Now, see this myeloid progenitor cell will be synthesized and then it can give rise to any kind of cell, including the RBCs, erythrocytes, platelets, and so on. So, and others common cell which can give rise to either eosinophils, basophils, mast cell, neutrophils, or monocytes and macrophages, right? So the question is, what is going to decide that which of the following cells should be synthesized? ठीक है, तो अब जो हम डिस्कस करने वाले हैं, हम कुछ केमिकल फैक्टर्स की बात करेंगे, कुछ को डिस्कस करेंगे. These are known as interleukins. Majorly, these are kind of chemokines which will regulate the process of hematopoiesis, right? So the synthesis of different types of cells, for example. So I'm writing few names. For example, who is going to tell that myeloid stem cells should be divided into either erythrocytes, platelets, or other type of cells? So first of all, there is interleukin 3, IL-3. All right. You don't know what it is not right. Interleukin 3. Right. And interleukin 6. Sorry. Interleukin 6. And there is granulocyte monocyte colony stimulating factor, CSF. All right. If these factors are present, so they will decide which kind of cell should result from the same myeloid stem cell. Okay. For example, this cell will give rise to eosinophil if here and another signal is present which is given by interleukin 5. So this interleukin 5 will tell that eosinophil should synthesized. Okay, so this interleukin 5 will responsible for the formation of eosinophil. And if basophil is, so there is no need for anything. And these these chemical or the peptides will decide if a basophil will synthesize, or this will lead to the formation of a mast cell. Now, if there is erythropoietin, which is a hormone responsible for the synthesis of erythrocytes, right? If there is a, so erythropoietin, a hormone, ER. So if erythropoietin hormone is there, it will lead to the synthesis of RBCs. If interleukin 11 is there, along with another hormone called thrombopoietin, so it will lead to the synthesis of platelets, right? So these factors are some of the very important factors which will decide which type of cell should result. If there is a neutrophil, for example, here, so there should be granulocyte CSF. So now you need to understand this. There are two versions of this. One is known as granulocyte monocyte, we have discussed earlier, which can lead to the synthesis of either granulocyte or monocytes. But if there is only granulocyte colony stimulating factor, it will lead to the synthesis of granulocyte. And you know, neutrophils are major type of granulocytes. So in presence of GCSF, neutrophils will result. And if there is concentration high for monocyte colony stimulating factor, so it will result into the monocytes, and monocyte will mature into macrophages in presence of interleukin 1, comma, 4 and 6. So I'm directly writing 4 and 6. So 4 and 6. Suppose there is a combination of interleukin 1, interleukin 4, interleukin 6. These monocytes which are present in blood circulation, they will go into the tissue spaces, and there they will be matured into macrophages. Macrophages are major type of phagocytic cell, whereas neutrophils are small in size and therefore, but they are also having all the properties which macrophages have. They are also a kind of phagocytic cell, right? So we will discuss these neutrophils in more details. We will discuss which type of enzymes and uh, granules they are having inside their cytoplasm. Similar granules are also present in macrophages, which will degrade the components of microbial cells, and they will engulf and digest these microbes, which can cause infection. So are you able to understand this? Okay, so I'm telling once again. So this myeloid stem cell is actually coming out of hematopoietic stem cell, and this hematopoietic stem cell, once synthesizing the myeloid stem cell, then okay, so there should be something, right, which is actually responsible for making this myeloid stem cell. So this is called SCF, stem cell factor, right? So the stem cell factor will help in the differentiation of this hematopoietic stem cell into myeloid stem cell. This myeloid stem cell, in the presence of interleukin 3, 6, and GM-CSF, which is actually the granulocyte monocyte colony stimulating factor, which is a combination of the two factors, granulocyte and monocyte colony stimulating factor. All right. So it will result into one of the following cells, but in the presence of certain other chemicals. For example, if there is hormone erythropoietin, so the erythrocytes will be synthesized. If there is thrombopoietin along with interleukin 11, so the megakaryocytes, which are the mother cells for platelets, will be synthesized, and finally the platelets, which are not the complete cells, but these are known as cell fragments, will be synthesized. If there is interleukin 5, so eosinophil will result, and in the presence of these granulocyte colony stimulating factor, neutrophils, and monocyte colony stimulating factor, the monocyte will be synthesized. Monocyte will come out of the blood circulation, and they will be differentiated into the macrophages when they are present in the tissues in the presence of some other chemical signals called interleukin 1, 4, and 6. Okay, so that's how these cells will result. Now, so many bacteria and other foreign particles that enter into the body, they are taken up and they are destroyed by the neutrophils in a process. This is known as phagocytosis, which is also a common process utilized by macrophages, right? So the phagocytosis, we have discussed about phagocytosis in our last class. So I will not go, we will not go into the details, but these neutrophils are performing this phagocytosis, right? Neutrophils respond rapidly to the presence of foreign particle. So once there is a foreign particle which is not belonging to our body, and if there is a danger that it can cause infection or harm to our body, it will be destroyed by neutrophils. The phagocytosis of bacteria by neutrophils is promoted by some opsins. Opsins are some proteins which will coat the bacterial cell or microbial cells, right? Because the neutrophils and the microbial cell both are having negative charge, so they will repel each other, right? So there is a requirement to coat the bacterial cell or microbial cell so that it can be engulfed by the or uptaken by the either neutrophils or macrophages by a process called phagocytosis. So the proteins, so who is going to perform this function? Opsonins. We have discussed C3b and C4b in our last class, and antibodies are also one major kind of opsonin proteins, and the process is known as opsonization. Means by coating the antigen by either antibodies, C3b, C4b, and finally uptaken by the macrophages is known as opsonization. So important option include antibodies, complement proteins, including these C3b and C4b, which are protein fragments or fractions. Neutrophils possess the receptors that bind to the foreign particles coated with these proteins and trigger their ingestion and destruction. So this is also very important. These neutrophils will possess some kind of receptors over them. So we are going to discuss these receptors also. So for example, which receptors are major type of receptors which can be present over these neutrophils. So first of all, look into this particular diagram so that you can understand. All right, so this one. So the major receptors found on the surface of neutrophils, the structures that bind to different molecules and their functions. So a neutrophil, which is the same cell as I showed you in this particular diagram, this one, is actually having different kinds of receptors over its surface, right? So this is a neutrophil. And this neutrophil is having different receptor types over its surface so that it can help in the binding of bacteria or some microbial pathogen, right? So the receptors which are present over the surface of neutrophils or these granulocytes include CD32, CD35, CD45, CD11C or CD18, CD11. Okay, so this 11 is actually A, B, and C along with CD18. So it is a dimer actually. So one is CD11A along with 18, CD11B along with 18, CD11C along with CD18. So first of all, what is the CD? CD is cluster of differentiation. So major CD types are characteristic of any cell which are present over its surface. So any cell, अगर कोई भी cell है उसको हम recognize करेंगे उसके CD markers की help से. So these CD markers which are present over this particular cell will decide or will differentiate different immune cells. For example, neutrophils are having these following CD markers: CD32, 35, 45, 11, ABC, and CD18, right? These are major receptors which are present over surface of neutrophils. Each receptor is having some specific function. So what is the function of CD32? This is also known as antibody receptor. So antibody will come and will bind to this receptor. How? Let's see. So this diagram, for example, there are two. So this Y-shaped molecule is antibody, right? So antibodies are coating this pathogen. Suppose this is bacteria. This is a bacterial cell. And once they are coating this bacteria So this is bacterial cell. These are antibodies which are coating the Y-shaped molecules. And see, this Y-shaped molecule is actually having two major reasons. One is known as fragment of crystallization, this arm, and the two arms are responsible for antigen binding. So they are called FAB, fragment antigen binding, and the lower region is called FC, fragment of crystallization. So this FC will bind to this receptor. This is known as CD32. CD32 receptor is also known as cluster of differentiation 32 or FC gamma receptor second. This is FC region receptor and gamma is written for IgG. So the major antibodies which can bind to the surface of neutrophils are immunoglobulin G, IgG. This FC region can bind to this receptor and it will make a complex called receptor ligand complex. And finally, these pseudopodia will come over this bacteria, and this cell, which is a neutrophil or macrophage, will engulf this coated bacteria. So this coating is known as opsonization, and eating is known as phagocytosis. So there are two processes. And these complement proteins called C3b or C4b, they are having different receptors. So these receptors are called CD35 or complement receptor 1. All right. So these receptor will have different functions. So two of them, two already we have discussed. So the third one is actually to zoom this one. All right. So here is the diagram for neutrophil, right? And it is showing that there are different type of receptors which are present over neutrophils. CD32 will help in binding with antibodies. CD35 will help in binding with complement fragments called C3b and C4b. Okay, still I'm not audible. Suprita is saying my voice is not coming. Guys, are you able to hear me? Am I audible? All right, so no problem. तो हम डिस्कस कर रहे थे, देखो जो न्यूट्रोफिल्स की सरफेसेस पे अलग अलग टाइप के रिसेप्टर या सीडी मार्कर्स होते हैं, उसकी बेसिस पे हम अलग अलग सेल को डिफरेंशिएट कर सकते हैं, क्लासिफाई कर सकते हैं. So major receptors or major cluster of differentiation which are present over surface of neutrophils are CD32, CD35, CD45, CD11A, 11B, 11C, right, and CD80. So these are major receptors. They are having different functions. First of all, we have discussed about CD32, which are responsible for binding with antibodies like this. So the CD32 will bind with the FC region of these antibodies. So these are specific for binding with antibodies. Similarly, CD35, which is also known as complementary receptor 1, so it will bind with complement protein, either C3b or C4b, which will bind to this CD35. There are other receptors also, with, for example, CD11ABC. So CD11A along with 18, this is a heterodimer. Okay, so it will bind with these molecules called ICAM1 or ICM2, right? And these are also responsible for, including this 11A, 11B, and 18, they are responsible for cell adherence. Okay, because these molecules are present over endothelial cell structure. If you remember the last diagram over page number 167, this one. If you remember this particular diagram, I told you that these neutrophils will first bind with these endothelial cells. Okay, then they will come out of blood circulation by a process called as extravasation, and then they will move towards the site of infection or injury. Suppose this is injury, this is some knife or something, thorn or, okay, so there are lots of microbial cells which can come along with this injury. So, what is the function of neutrophils? Neutrophils should reach at the site of injury, right? So, these neutrophils will come out of blood circulation. So, first step is to bind these cells to the endothelial membrane, or these cells which are making these blood capillaries or blood vessels, right? So, they will help in this binding. Now, see this particular diagram. These molecules which are present over neutrophils, including CD11A and CD11B along with CD18, they will help in cell adherence. They will help in sequencing out or extravasation coming out of these cells from the blood capillaries. So they will bind with endothelial cell structures, and then they will move between the two cells. As you can see from this diagram, they will move in between two cells. So this is endothelial cell, this one is also endothelial cell, but this is neutrophil. So this neutrophil is coming out of this blood vessel in between two endothelial cells. So first of all, it should bind with the membrane so that it can come out of blood vessel. I hope this is clear now. These molecules will help in cell adherence, and then it will come out of blood circulation. Therefore, it is able to reach at the site of infection very early. These are the first cell types which can reach at any site of inflammation or injury or infection. Now, there are other molecules, receptors, for example, CD11C and 18, which will bind with some complement fragments, but it is unknown. We do not know which type of molecule will bind with these receptors. Okay, not very clear. Then there is CD45. Some cell membrane enzyme will bind with these receptors, CD45. So these are not very significant, right? Major significance to the neutrophil are of CD32 and CD35, as you can see in this diagram also. So CD32 is helping in binding of Fc region of antibody. These are antibody receptors, CD32, and CD35 are complement receptor 1. So this will help in binding with this complement protein C3b or C4b. All right. So we were discussing this. Neutrophils as receptors that bind with foreign particles coated with these proteins and trigger their ingestion and destruction by a process known as phagocytosis. Neutrophils kill ingested bacteria by means of reactive oxygen metabolite generated through the respiratory burst, and as well as through the activities of lysosomal enzymes. Right. So already we have discussed this one. If you do not remember, telling you the page number 151 of your QA. So if you go to page number 151, you are able to find this particular uh, diagram, or whatever you call flow chart. So these are reactive oxygen species, these are reactive nitrogen species, how they are synthesized. So the reactive oxygen species are the product of oxygen itself in presence of some enzyme called NADPH phagosome oxidase enzyme, or simply NADPH oxidase. This oxygen will be converted into, okay, so this is an oxidase, right? So what will happen, this NADPH will be converted into NADP positive, and this H positive or one electron will be donated to this oxygen. so that a net negative charge will come, and this is known as superoxide anion. Right. This superoxide anion is more reactive, and it can bind with or react with the plasma membrane. Then in presence of superoxide dismutase enzyme, this superoxide anion will be converted into a more stable form that is known as hydrogen peroxide, H2O2, which will further degrade it into hydroxy radical. Or so the hydroxyl radical can be of two types, either a single electron or a complete negative charge, which is due to two electrons present over this oxygen molecule. Lone pair of electron, right? So if a halide, for example, chloride or chlorine, chlorine, bromine, iodine, something will come with a negative charge in presence of another enzyme, myeloperoxidase, this hydrogen peroxide can be converted into a microcidal or bactericidal compound. This is very toxic. So if the chlorine compound will come, Cl negative will come, it will be converted into hypochlorous acid. So this is an acid, it will have a lower pH, and this will be very harmful to the cell if it is bacteria or some other microbial cell which can cause infection. It will try to kill that particular microorganism. So this is bactericidal, right? It will react with plasma membrane very fast. Similarly, reactive nitrogen species, which are also very harmful in the cell, are produced. So this has been asked several times in many competitive examinations. You will find that this arginine amino acid, right? Arginine is denoted by a letter. This is called R. Arginine is R. Because we cannot represent this amino acid with A, because A for alanine, so we have given the second letter R. So R is the arginine amino acid, will be converted into nitric oxide, sorry, L-citrulline, and when this L-citrulline will be synthesized, nitric oxide will release. This nitric oxide is a kind of reactive nitrogen species which can react with plasma membrane or other lipids present in the membrane. This nitric oxide will be synthesized by an enzyme. This is called inducible nitric oxide synthetase enzyme. Sorry, synthase. There is a difference between synthase and synthetase. So synthase is not requiring ATP energy, whereas synthetase do require energy source, for example, ATP or NADPH. Right. So this nitric oxide, now this, if in the cytoplasm superoxide anion is present already, this nitric oxide can combine with the superoxide anion, and it will lead to the synthesis of an harmful intermediate. This is known as peroxynitrite, which will be further converted into S-nitrosithiol or nitric this dioxide, nitrogen dioxide, NO2. Right. So all are, including nitric oxide, nitrogen dioxide, and peroxynitrite, all are reactive nitrogen species. And in fact, this is a combination of reactive oxygen and reactive nitrogen species called peroxynitrite, right? Because this is coming out of this nitric oxide and superoxide anion. All right, so this we already have discussed. If you have any query, you can please ask. All right, so we should move to some another PDF, right? Then the fourth point is neutrophils destroy ingested material and do not process it for the immune system. So neutrophils are different in sense because they will not try to activate, they will not process, they do not process the antigen or the material, foreign material, so that immune system can recognize it. As a result, it will not provoke an immune response. So once a neutrophil is able to kill a microbial cell or pathogen, it will not activate the immune response, a fully pronounced immune response. There are other cells also, for example, eosinophils. These are also very important, and question has been asked. Eosinophils are specialized phagocytic cells. So these are also having this phagocytic role, right? But they will destroy the parasites. Parasites in the sense, for example, helminths. So it is given actually somewhere, 34, I think. So if you go into the details of eosinophils, they are also having the same structure, but they are specialized phagocytic cells because uh, where it is given actually. All right, so here, eosinophil like neutrophils are motile phagocytic cells. They are also phagocytic cell and they do move, they are motile, and the movement is crawling actually. They are having crawling movements that can migrate from the blood into the tissue spaces, right? By the same process, extravasation and then chemotaxis. This phagocytic role is significantly less important than that of neutrophils. And it is thought that they play their most important role in the defense against multicellular parasitic organisms. So they are very specialized in killing the multicellular parasitic organisms, including some worms, right? So if there is a worm infection or worms are present inside our body, these eosinophils will work against these multicellular organisms because these are kind of eukaryotic microbial. These are not microbes. In fact, worms can have size which can be seen by the naked eye. So these eosinophils are very, very important because they can even kill multicellular parasitic organisms, including some worms. They can be found clustering around the invading worms whose membranes are damaged by activity of proteins released by the eosinophil granules. So these are having some very potent chemicals, some proteins which are stored inside the granules in the cytoplasm. So let's discuss what kind of granules they actually have. So eosinophils are having granules, so in which they can store cationic proteins, which are having some positive charges, right? For example, EPO, ribonucleases, which can degrade the RNA. So if you are able to see, ribonuclease is not present in any one of them. They're having some cytokines, chemokines, and so on, right? IL-4, IL-10, IL-13, and so on. All should not. So these cationic proteins, which are present inside the cytoplasm or granule, they induce the formation of reactive oxygen species, and these reactive oxygen species can be secreted out of cell to kill the multicellular parasites, right? So cytokines, for example, they are having antiviral activities. They are responsible for killing the parasites. So in examination, generally, question related to the parasite is asked, and always you need to remember that eosinophils are cells which are responsible for their removal or destroying them, right? So this one is important. All right. So we had discussed eosinophils, then the basophils are granulocyte and that are found in the low number in the blood and they participate in some allergic reactions. So if you talk about basophils and mast cells, which are also found from basophils, they are taking part in allergic reactions because they are having some molecules which they store, right? So see this diagram, this table first, so that I will tell you. So the basophils and mast cells, They do have histamine, and histamine is a kind of molecule which help in vasodilation and smooth muscle activation. Also, they have some cytokines, interleukin 4 and 13, then they can modulate the adaptive immune responses, which the neutrophils are unable to do so. So these are mast cells and these are basophils. See this difference. So they are almost similar. There is very little difference between the two cells, but if you see this, some differences in the nucleus, right? Shape of nucleus. Now they will release some molecules. They will have some stored granules which will help in allergic reactions. So we will discuss this. First of all, the basophils. So basophils are non-phagocytic granulocyte. They cannot perform this phagocytosis. So you need to remember this. They are non-phagocytic. That contain large granules filled with basophilic proteins. They stain blue in the standard this stain. So therefore, because of this blue stain, they can take the blue stain. They're called basophils. Eosinophils because they are red stain, and neutrophils because they are neutral. They cannot be stained by the same staining procedure. Therefore, they are called neutrophils. So actually, name is depicting their features, right? So basophils are stained blue, and therefore the name. But these are non-phagocytic cells or granulocytes. They do have granules in their cytoplasm, but they cannot perform the phagocytosis, right? Basophils are relatively rare in circulation. Number is very less in comparison to neutrophils. Neutrophils are major cells found in WBCs. 60 to 70% of all WBCs are neutrophils, but basophils are hardly 1%, less than 1% in fact. Basophils are relatively rare in circulation, but can be very potent in response to the binding of circulating antibodies. Basophils release the content of their granules. Histamine, for example, histamine, one of the best non-protein on basophilic granules, increases the blood vessel permeability and smooth muscle activity, and this can result into an allergic response, right? Critical to our response to the parasite, particularly helminths, but in areas where worm infection is less prevalent, histamine are best appreciated as cause of allergy symptoms. Okay, so major function of these cells are related to allergy. Then there are mast cells are released from the bone marrow into the blood in an undifferentiated cells or state. They mature only after they leave the blood. So once they leave the blood and come into the tissue spaces, they will be differentiated and they will mature. Mast cells can be found in a wide variety of tissues, including the skin, the connective tissue, various organs, mucosal epithelial tissue of the respiratory. Okay, so all the systems, all the tracts, digestive tract, urinary tract, all these tracts do have these cells. Like circulating basophils, these cells have large number of cytoplasmic granules that contain histamine. So because they are also having the histamine molecule, and this is one of the allergic molecule which can cause the allergy, right? So, and other pharmacologically active substances. Cell These mast cells can play an important role in the development of allergies. So along with basophils, they are major cells which are responsible for allergy uh, reactions, right? Some scientists also have proved that the basophils are blood-borne version of mast cells. So in the blood, they are called basophils, but if they went into the tissue spaces, they will mature and they will become mast cells. Mast cells, right? Other cells figure the function. All right. If you have a query, please ask. So all the phagocytes, including the neutrophils and macrophages, they are having four basic steps to kill any pathogen. So first step is chemotaxis. Chemotaxis is, as I told you, the crawling movement towards the, suppose this is a bacterial cell, so it will release some chemotactic signaling molecules in response to which these neutrophils will come towards this, so that this process is called chemotaxis. The movement of immune cell in response to the concentration gradient of some chemicals or chemokines, right? Then there is adherence. Adherence, as I showed you the previous diagram, so this one. So this is called adherence. Adherence is with the help of antibodies and some complement proteins. They will coat this antigen or bacteria, and because there are some specific receptors which are present over the cells, for example, neutrophil to have CD32 and 35, they will bind together. This process is called adherence, right? So first of all, we have discussed about chemotaxis, which is the movement of these immune cell towards the chemical gradient, right? Second is adherence. This bacteria will adhere to the cell surface with the help of some, this is a coated bacteria, right? With the help of antibodies or complement protein, it will bind to the cell surface receptors. So there is a query also. It is the nucleus that takes stain, and it is the same in every granulocyte. Okay, so no, actually, so basically these cells which are called neutrophils, basophils, and eosinophils, they are categorized depending upon staining, right? So I'm trying to share one more window, so if I can. So, okay, so I'm showing you one more diagram. Wait a minute. Okay, this one. Are you able to see this Chrome page? I'm showing you one more image. So if you are able to see this, this Chrome page is visible to you all. Student, guys, please confirm so that I can explain this to you. All right, fine, because I do not know whether you are able to see this or not. Okay, so see this diagram, right? So it is clearly showing that the nucleus is same stain, because but they are having differences in staining of these granules. Why they are called granulocyte? Because they are having granules inside their cytoplasm, right? Nucleus is one thing, but they are having some granules which are called primary and secondary granules. So if you see this, basophil will be stained blue because they are having some different kind of granules. Eosinophils, they are stained red if you're able to see these red small granules inside their cytoplasm. And neutrophils actually do not take any stain, not blue, not red, therefore they are neutral, and therefore they are called neutrophils. Are you able to understand? This is not the nuclear staining. Actually, we are staining the cytoplasmic granules. We are staining the cytoplasm. All right. If you are able to understand this, then come to this particular diagram back. So who has asked this one? So Shubhrata, are you able to understand this? We are not staining, okay, nucleus. We are staining the cytoplasmic granules. And in today's class is interesting because we are going to discuss about the granules also. I will cover that particular topic. We are moving to this portion, right? We are coming to that. All right. So there are four steps in phagocytosis. First is chemotaxis, in which the immune cell, for example, neutrophil, will move towards the bacterial cell and it will try to attach it with the help of some chemical, with the help of some antibodies or complement proteins as shown in this particular diagram. So these are antibody molecules, Y-shaped molecules are antibodies, and these goal, round are called C3b or C4b. These are complement proteins, right? So they will bind with these receptors, either with CD32, antibody will bind to the CD32, and they will bind, these complement protein will bind to CD35. CD is actually called cluster of differentiation, right? So these receptors are present over neutrophils, so they will bind with the bacteria coated with antibodies and other molecules, right? So this is called opsonization, and then we are talking about the phagocytosis. Third step is, once it is attached to the neutrophil surface, bacteria is uptaken inside the cytoplasm, and what will happen, the pseudopodia will come out and it will enclose this bacteria into a vacuole like structure called phagosome, right? So this process is called ingestion. So the cell engulfs the organism, that is the bacteria, within its cytoplasm, and it will be killed ultimately, which is called the digestion process, right? So digestion, what will happen now in the last step, because there are a large number of cytoplasmic granules which are present inside the cytoplasm of this neutrophil. These are called primary and secondary granules. They will join this particular phagosome. They will release its enzymes, contents. Lysosome will release the acidic hydrolases inside the same phagosome, and they will make a structure called phagolysosome, right? Right. So it is also written here, phagolysosome will form, and ultimately all the components of the microbial cell will be disintegrated, will be digested, thus killing the microorganism, right? So these are four basic steps. Hope you are able to understand this. This is very clear. We have discussed this diagram many times, right? So then, sorry, so I should show you. Right, so there are a few lines. We will go step by step. So bacterial invasion and the tissue destruction result into the production of many chemotactic factors. So one is the very important chemotactic factor. So why we are discussing this chemotactic factor C5a here? Because if we are discussing this chemotaxis, right? So this bacteria this is a single cell shown here. But in fact, there are large number of bacterial cells which are present in some tissue spaces, right? देखिए बहुत सारे बैक्टीरिया होंगे तो कंटिन्यूसली टिश्यू को डैमेज करते रहेंगे. टिश्यू डैमेज होगा तो क्या होगा, एक मॉलिक्यूल प्रोड्यूस होगा जिसको बोलते हैं C5a, which is a complementary protein. I told you these complementary proteins are produced by liver and they are present in the blood in an inactive form. So they will be activated. They are zymogens actually, which will be activated in response to some other signal. So it will be cleaved. C5 will be cleaved into C5a, which is a small fragment, and C5b, which is the larger fragment, right? And the C5a, which is smaller in size, will act as a chemotactic factor. Chemotactic factor in the sense it will attract the neutrophils towards this site of infection, this bacterial site, right? So one thing is C5a, which is very, very important, which is a kind of chemotactic factor. And second is this interleukin 8. I told you about various interleukins. in the hematopoietic pathway, but interleukin 8 was not there, right? So this interleukin 8 is important because this is also an important chemotactic molecules, which is a small protein. Uh, Interleukin 8 is produced by many cells, macrophages, fibroblasts, epithelial cells, endothelial cells. They all can produce the interleukin 8. Small lipid soluble molecules such as leukotriene released by the blood platelets can also attract the neutrophils. So all these C5a, which is a complement protein product, interleukin 8, which is produced by the macrophages, fibroblasts, epithelial cells, endothelial cells, which are present around the site of infection. They can produce these molecules and they will attract the neutrophils, so that neutrophil will come out of the blood vessels and they will reach at the site of inflammation or injury, right? 167 page number. So they will come out of this blood vessels or circulation, and they will, by the crawling movement, by the process of chemotaxis, they will move. But who is going to tell them कि कहाँ जाना है? So C5a, interleukin 8. Okay, so these molecules are actually telling them where to go. So in their concentration gradient, they will start moving, and where is the maximum concentration? They will reach at the site of inflammation or injury. All right. Then some few lines are also important in this paragraph. So invading bacteria release peptide containing a unique amino acid, which is formilated methionine. If you already have studied the molecular biology, you should know that in prokaryotes, in bacteria, this is the main difference between a prokaryotic cell and eukaryotic cell. They are having this formilated methionine. And this formilated methionine is a kind of tag, right? So it will also recognize that is not produced by mammalian cells, and therefore neutrophils are strongly attracted to the peptides which are actually having this formilated methionine. All right, so this is a kind of molecule which will attract these neutrophils so that they can reach at the site of injury. As a result, move rapidly at the site of bacterial invasion, right? All right, so they will crawl and they will reach very rapidly. So there are two types of cytoplasmic granules, and you should know that once the neutrophils are synthesized by the process of hematopoiesis, as I showed you in the previous diagram, right? So this one. So please see this again, and what I want to show you. So suppose these are neutrophils, right? So once these are produced, they will not divide. They will not able to divide to make new cells, right? So there is no mechanism by which they can divide. Therefore, they are having some fixed life. The chromatin in the nucleus is compacted and assume this segmented shape since these cells are no longer able to divide. They cannot divide. Second thing is they are having two type of cytoplasmic granules. अब यहाँ पे इस डायग्राम को एक मिनट सॉरी all right, so here there is some problem in the PDF. So try to understand this. So if you are able to see this one, there are two type of granules which are present in the cytoplasm. One is the dark primary granules. These are called primary granules. And there are some pale color secondary granules. So these are lighter in color, but they're called secondary granules, right? So they are different because they are having different set of enzymes. The primary granules, which are dark in color, electron dense structure, contain bactericidal enzymes, including myeloperoxidase. Where we have studied myeloperoxidase into the diagram where we were these reactive oxygen species synthesis, right? Page number 155 in your tubing. Sorry, 151, I think. So myeloperoxidase, here we have studied, which will convert hydrogen peroxide into an dangerous, or you can say bactericidal compound, hypochlorous acid. So these primary granules have this enzyme, myeloperoxidase, first, and lysozyme. Lysozyme is also responsible for degradation of bacterial cell wall, peptidoglycan cell wall. So both enzymes are very harmful to bacteria and they're present in primary granules, right? Neural protease such as elastase, acid hydrolase, and beta glucuronidase, cathepsin B, all these are present in primary granules and they can degrade elastin. Okay, so acid hydrolases can degrade some other components, and cathepsin B, which are bactericidal in nature. Then there are secondary granules, which are not. Sorry, so something is missed. So I'm reading this, what is written in this particular. So there are. All right, so secondary granules, which are not electron dense, containing enzymes such as lysozyme and collagenase, and some iron binding protein called lactoferrin. So, okay, so I'm trying to write it here so that you can understand. So the secondary granules also do have lysozyme. So you will not confuse primary and secondary granules both have lysozyme, right? So this will not, yeah, then collagenase to degrade the collagen, because animals do have collagen, right, in connective tissue. And there is iron binding protein, Fe binding protein, lactoferrin. All right, so these are some molecules, LACT, lactoferrin, right, which are found in the secondary granules, which are not very electron dense structures, which are pale in color. They have lysozyme, collagenase, and iron binding protein lactoferrin. Now let's discuss their functions, right? So in the next page, this is a neutrophil and it is having two type of granules. So the dark granules are called primary granules and the light granules are called secondary granules. Both are having lysozyme. So you are able to understand up to here that lysozyme is present in both type of granules, which will destroy the bacterial cell wall, peptidoglycan cell wall. But primary granules do have myeloperoxidase enzyme, which is very important for production of some reactive oxygen species, right? And the process called respiratory burst, right? So the process by which the oxygen reactive species or the reactive oxygen species are produced is called respiratory burst. So myeloperoxidase enzyme is very, very important in this pathway. Then they are having some neutral and acid hydrolases, which will degrade the bacterial proteins. Hydrolases, which will degrade some peptides and proteins which are present in the bacterial cells. There are some cationic proteins in the primary granule which will kill the gram-positive bacteria directly. Then there's iron binding lactoferrin protein which bind with iron, and therefore if the iron is sequestered, so it will prevent the bacterial growth, because bacteria or the microbial cell do require iron for their growth. If the iron is not free, not in free state, this lactoferrin will bind with all the iron. Okay, so it will not allow the bacterial cell to grow. Collagenase, which will degrade the connective tissue in animal, because they are all the animals do have this collagen. So collagenase is an enzyme which will degrade the collagen. So because they are having two type of granules, primary and secondary granules, they will damage. All right, so see this diagram now. Okay, so I told you this is the neutrophil plasma membrane, right? So this neutrophil cell, we can suppose this is a neutrophil cell. If you are not able to understand, suppose this is a neutrophil cell, right? Which is a phagocyte. So it will have two type of granules. One is dense structures, which are actually primary granules. One are lighter structures, which are actually secondary granules, right? So they are having large number of these granules. Now see this. Once some bacteria is bound to the membrane of neutrophil with the help of this receptor, which is known as CD32. Let's remove all this, because up to now you are able to understand this is neutrophil, right? So it is having two type of receptors, as we have discussed. One is called CD32 and another is CD35. So here we are discussing CD32 because this Fc region of antibody is actually binding with the antibody receptor. This is called CD32, right? This is antibody. This Y-shaped molecule is antibody, which is already bound with this antigen or this bacterial cell. Antigen, right? So this antigen is bacteria here. This is a bacterial cell, suppose. Okay, so it is also written here, this is a bacterium. What will happen? This NADPH oxidase, which is an important enzyme, as we have discussed in the reactive oxygen species formation. NADPH oxidase is the first enzyme which will convert the oxygen into the superoxide radical or this anion, right? Then superoxide dismutase and myeloperoxidase will convert into the hypochlorous acid. So see this, inside the cell cytoplasm, there is lots of NADPH present. This is the reduced power, right? So this will be converted into NADP positive, and the electron will be donated to the oxygen molecule so that it will be converted into the superoxide anion. This anion will be converted into the stable, in presence of water molecules, converted into the H2O2, which is called, what is this? Hydrogen peroxide. Hydrogen peroxide, then in presence of myeloperoxidase, which is actually present in the primary granule in these neutrophils, will release outside the cell, and it will convert this hydrogen peroxide into, in presence of some chloride ion or some halide ion, into the hypochlorous acid, which is bactericidal. So once it will go inside the bacterial cell, it will kill this bacteria. So that's how this pathway is controlled. It is coming, this myeloperoxidase, as you can see this arrow, it is coming actually from the primary granules, which is present inside the cell cytoplasm. So from the primary granule, myeloperoxidase is coming. This myeloperoxidase is converting into a harmful bactericidal compound called hypochlorous acid, and this will kill this antigen or this bacterium. So that, all right. So that's how neutrophils are able to kill the bacterial cells by the process of phagocytosis or by releasing their enzyme in the surrounding environment so that they can kill the large amount of bacteria, large number of bacteria. If you have a query, please ask me, because we have discussed everything about neutrophils and their cytoplasmic granules, what kind of enzymes they have, how these enzymes are operated so that they can synthesize reactive oxygen and nitrogen species out of oxygen and arginine amino acid to kill the microbial cells. And most important, they will not activate immune system. So they can kill the microorganism, they can kill the bacteria, but they will not stimulate, they will not activate the immune system. But macrophages, yes, they will activate the immune system also, because macrophages will act like antigen presenting cells and they will activate some other branches of our immune system, so other type of cells, including the B and T cells. Right. If you have a query, please ask. We will discuss further in our next class. So I will start. In the next class, we will discuss about epitopes, haptanes, haptane carrier conjugate, and all these things. All right. And I will tell you how many amino acid and carbohydrate molecules are required to activate an immune response. So at least to make an antigen, what should be the minimum size? So if you increase the size, if you increase the complexity, if the foreignness is increased, so and if the stability is well, ultimately the antigenicity will increase. So there are two terms, antigenicity and immunogenicity. Now you need to remember that all antigens need not be immunogens. So immunogen means an agent which can activate the immune response, but antigen can be anything. Any foreign molecule can be an antigen, but for an immunogen, so सभी antigen immunogen हो ऐसा जरूरी नहीं है. But all immunogens are antigens. So you need to remember this like this. So all immunogens are antigens, but all antigens need not be immunogens, right? So you need to remember this. Okay, all right. If you do not have any queries, that's all for today. We will continue our class. So today only we have discussed about phagocytosis, how neutrophils are able to do phagocytosis. We have discussed, and that's all for today. If you have a query, you can ask here, or if you're not able to ask right now, please go through this particular content I will share soon, and then please ask your queries. All right. So that's all. Thank you. Good night.